Hi guys, I'm going to draw what we call a graphic organizer, and a graphic organizer is basically only one sheet of paper and has all the ideas on there. So here's what I've made to make sure it fits on one sheet, and you can see it's about a fourth, and then I think we have a, I don't know, this is a bigger one here, like an L, and then a small one there. So that's what you're going for in the end. Okay, so let's get to it. So we are doing proteins, so we want to write that down. And I'm doing this so that you can write it with me, not just watch it. You just watch it. I feel like that's not very useful. So what are the elements we find in proteins? Well, carbon, always for the biological molecules. Hydrogen, always. Oxygen, always some of that. Nitrogen, not surprising. And then the last one here is sulfur. So that one is unique to proteins. It's in the amino acid called cysteine. It has a sulfhydryl R group. You'll see what that means in a minute if you haven't read already or looked at the other lectures. And so you can actually mark, if you, let's say back in the day, they used to use radioactive sulfur. And so they could track where proteins were located because they were the only ones that would have the sulfur in them. And so wherever the radioactivity was, that's where the proteins were located. Okay, then I want to say, what are our functional groups? It's those groups that give characteristics to molecules. So proteins always have two groups. They have amino groups. I'm going to track the amino groups in red. Doesn't matter what color you use, but you can pause this actually and go get 3 different pens and a pencil, colored things, but I want you to be able to see it visually as we go through, because you'll be able to see what the red is, see what the blue is, etc. So amino groups, as you know, are the ones with the nitrogen, and in aqueous solution, meaning water, which is what the cell is, they go back and forth between picking up a hydrogen and having a positive charge and dropping off that hydrogen and being neutrally charged or no charge. So I could have a picture of amino acid, well, let's just go an amino group, and it could look either way, like this. These would both be amino groups. Okay, the second group that's in all proteins is the carboxyl group. Boxcar, that's the choo-choo group. C double bonded to an O, bonded to an OH, and this one also goes back and forth, but this is acting instead of as a base like the amino group because it picks up hydrogens. This acts as an acid because you can see, right, the hydrogen was lost, it's here, and then it's not there, and that's why there's a negative charge. We need to know the monomer, single unit of proteins, so that is amino acids. There are 20 types, and some organisms, like certain bacteria, have modifications on those, but generally we think 20 types. And the R group, the variable group, which we'll see in a moment, is really important. That's what makes each amino acid unique, all 20 of them. All right, so that's the monomer, the building block. What's the polymer? Meaning the building blocks put together in a long chain. So the long chain of amino acids is called a polypeptide. Poly meaning many, peptide is having to do with proteins typically. And that's all I'll say about that for now. And those polypeptides are going to fold into different structures. So shape is really important for proteins. They're highly varied in shape, so much more varied than any other group. And that's why they have more functions. So I'll just write for the moment that there are structural levels. You'll need to know that. For the proteins, there's 4 of those. And then not functional groups, but functions, as in what they do. There are many functions of proteins, and I'll write down some of them. So one of the most important ones is acting as an enzyme. So enzymes are proteins that promote reactions, very specific because they're all particular shapes. Okay, we have transporters. Transport proteins are moving things either around the cell or around the body if it's a multicellular organism. We'll see all these groups we'll see throughout the semester. So just getting them now in our heads a little bit. Proteins can store energy to be used later. They can signal. Signaling allows the cell to communicate with the environment and to get signals from the environment or other cells. We have structural roles. They could be part of the cell itself or part of a multicellular organism's body. We have gene transcription regulation or gene expression. We'll say that for now. Or maybe we should just say gene regulation. Let's do that. And then let's write this down. You don't need to know this word yet, but when you're studying for your final, I want you to have the things we're looking at in the future, some of them down here. So you're going to see those are called transcription factors, or that's at least one type of gene regulator. All right, we have defense proteins. For example, antibodies. And the last one I'm going to list are contractile and motor proteins. Help things move around the cell or large organs like muscles, crawling, contractile, and motor proteins. They work together. And you'll see in the next unit, when we talk about the cell, these are going to be the cytoskeleton elements. Okay, so then on your page, you want to draw your line down, separating that off. Yours, of course, will be like right next to the edge of the page. And then I will zoom back in again. And now we'll look at the monomer of the proteins. That's the amino acids. So let's draw an amino acid. You'll need to be able to recognize these. So we've got a carbon attached to a hydrogen. Then we've got our two functional groups. We have the one with the nitrogen, which is your amino group. And remember, we're tracking those in red. And then we have our other group, the carboxyl group. And we're tracking those in blue. Those are always present. All right, and then we have a variable group attached to the top. Of course, carbon has to have 4 bonds, and so this, call it an R group, which is not actually an R element or something like that. It's can be a variety of things. So let's look at that. So R is your variable group. And that is what makes each amino acid unique and gives it its character. So because you know bonds and how the bonds, polar, nonpolar, charged, you understand how those bonds work, you'll be able to predict any amino acid's behavior pretty much. Our group examples. Circle I'm drawing here is what we call the backbone of the amino acid, so it's everything except for the R group. So all this is now represented by a dot. So let's say we have some groups with CH3 on them. So the carbon would be attached and the hydrogen's attached to the carbon. What about some groups with carbon-based rings on them? So I'm going to draw a hexagon. All the corners of that are carbons, and I'll just draw some double bonds in there. So both of these are carbon-hydrogen rich, right? Lots of carbon-hydrogen bonds, and as you know, that means that they are nonpolar because they share electrons equally, which also means that they are hydrophobic. And in the protein, you usually find them away from water. Okay, what about some amino acids with a hydroxyl group on it, the OH? Maybe we have a ring and then a hydroxyl group on top of that. Those, as you know, hydroxyl groups are polar. So we've got the OH bond, polar, which means it's hydrophilic. Which means it's going to interact well with water and would likely face towards it. So on the outside, I mean, there's also polar amino acids on the inside as well, but they'd be the ones bonding, hydrogen bonding with the water. They can also hydrogen bond with groups that are opposite charges, whether it's partial charges or whether it's a complete charge, which we'll see next. We have amino acids with amino groups on them. So this is not the amino group on the backbone. This is another amino group on as the R group. And this is positively charged. As you saw before, it can oscillate between those two. So we have positively charged. You'll see amino groups for that. And then we also have carboxyl groups that can be on there. Let me draw it without the hydrogen, so it's in the form where it's already dropped those off. So this is negatively charged. And you'll see it with carboxyl groups. Okay? And those guys could interact, and because they're totally positive and totally negative, they would ionically bond, like an ionic bond. We have hydrophilic and hydrophobic. We have hydrogen bonds happening, and we've got ionic bonds happening. So all these interactions between our groups within the protein and to the environment, that's what dictates what proteins can do, what shape they'll be, who they interact with or not. Really, 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 really important. Now let's look at the structural levels, which will also include looking at the polymers. There's always 3 structural levels, sometimes 4. So the first structural level, the primary, is based off of 2 amino acids and then a lot of them attaching. To begin, let's draw 2 amino acids attached to each other. So here's one, and then we have some sort of R group, which we just talked about that means. And it's attached. So remember, this is dehydration synthesis, which you learned about in a different chapter. Dehydration synthesis is what builds the polymers. And so what that means, dehydration, or you can call it a condensation reaction, you've lost a water. So the water was from the end of the carboxyl. So we're looking at part of it coming from this and part of it coming from the next amino acid's hydrogen. So the H and the OH come together, and then that means that they leave as water. I'm going to draw my second one to show you here. This is the second amino acid. And then we have the bond in between the two. And you can see that's where the water left. This is one amino acid. This is another amino acid. The bond in here has a special type of name for amino acids and proteins. What they're attached is called a peptide bond. And that's the one in between. So I could give you a picture and ask you to identify where the peptide bond is. What you're looking for is each amino acid has a nitrogen, carbon, carbon. That's one amino acid. Nitrogen, carbon, carbon. That's another amino acid. So just look for your nitrogen, carbon, carbon, and the bond in between the nitrogen, carbon, carbon is your peptide bond. And this part here, the non-R part, is the backbone. I mentioned that term, meaning it's the same in every amino acid, and it's the part that makes the long string. First level is called primary structure. Now I'm going to draw amino acids represented like this. We're looking at amino acids in a very long chain, many, many, many more. It's the polymer, the polypeptide. That's the structural level. This is the peptide bond. The big deal about this is it's a unique order or sequence of those amino acids. So who is connected to who? All proteins have different ones, and that's what makes them behave differently. Secondary structure. That long chain of amino acids folds into structures that are like curlicues, which is a helix. Call them alpha helices. Alpha helix. Helices is plural. Helix is singular. And then we have, this is really bad drawing. You can look in your book for much better drawings, but it's basically the ribbon of amino acid goes back and forth, and it bonds to each other between the curves. And we call that a beta pleated sheet. And then you'll notice I drew, after that, here's another alpha helix. There are some regions that we call disorganized, particularly at the ends of proteins, the polypeptides, and they're important as well, but these are alpha helices and beta pleated sheets. These are forming through hydrogen bonds between the backbone groups, not the R groups. Tertiary means third. Sort of sounds like tri, which you probably know tri means three. Okay, this is really when we get into the unique proteinness. This is when we get the 3D shape. And of course, as you know, structure allows function. And this is all dependent on the R groups and what order they're in. I'm going to draw an alpha helix, draw something like this, and then a loop, another alpha helix, far enough away where I can write something in between those. And then a beta pleated sheet, and another alpha helix over here, and disorganized end. Great. So pretend this is 3D and much more complicated. All right, so now this is not backbone, it's the R groups holding together. You can have R groups that are carbon-hydrogen type groups. And let's say you had a number of CH3s. They would probably be folded inside. And those interactions between those, London dispersion forces, or we can call them hydrophobic interactions. It's not a bond, but it is an association. And let's say we have a partially negative charged group, and over here we have a partially positive charged R group. Got partial charges in there. Usually that means a hydrogen's involved in biology. So we draw a dotted line for that. This is our second type of bond, hydrogen bonds. They're really important for holding it together. What about if you have an R group that's a complete positive and you have an R group over here that's a complete negative? They may fold and form an ionic bond. And then we have, I mentioned that proteins have sulfurs in them, and that cysteine is the amino acid with sulfur. So it has a sulfhydryl group, SH, but you're going to see that we need a bond. This one has an SH. When they come together, they lose the H's, and they can actually form a covalent bond. The rest of these are weak weak types of interactions, weak interaction, weak interaction, weak interaction relative to covalent bonds. But there are some between these cysteines if they're in the right place. And we call this a di-2 sulfide for the sulfur bridge because it connects them. And after tertiary structure, some proteins come together or some polypeptides come together to make the actual protein. So that's the fourth level. We're going to call that quaternary. So you have, what is it, more than one subunit. And you need all those subunits to come together to actually make a protein. When they're just the subunits, they don't count. Okay, so let's look at a representation of this. So I'm going to draw one in my black color. So we've got some alpha helices, we've got some beta pleated sheets, we've got it held together in a 3D structure. So there's one subunit. I'm going to draw a second subunit in a different color. So second subunit. And then I'm going to draw a third subunit that is folded in a different way. Okay. But you can see that I'm drawing them to where they're interacting with each other. And that's what holds it together. So once we get all that together, now this is considered a protein. The bonds that hold those subunits together are similar ones to the tertiary bonds. All right, now we have proteins, whether it's a single subunit that acts all by itself, so it is a protein already, or whether it's multiple subunits that come together to make a functional protein. It's doing its job, and it has to have a specific shape. So what happens when it loses that shape? So it can either lose it from the tertiary structure or the quaternary structure. I'm just drawing this in the bottom corner. So we have the third and fourth levels, and let's say they get disrupted and go back to the primary level, primary structure, that polypeptide. So how would that occur? There's interruptions of the weak bonds, hydrogen and ionic bonds primarily. So that process there of unraveling is called denaturing. So the protein denatures. They can fold back up in certain circumstances, and so we call that renaturing. What kind of conditions can interrupt weak bonds? So heat, and that literally is just because the molecules start shaking too much and the bonds come apart. You've got to think, though, too, that there are some organisms that live in hot springs and the geysers on the bottom of the ocean. So obviously their proteins are shaped in a way to where the heat does not denature them. So it's not like hot is always bad at all. It just depends on the organism, right? How those proteins are evolved to allow them to live in a certain environment. What else? We have pH changes. The delta, this little triangle, is an uppercase delta. That means change. So pH changes, and why is that? Because of those ions. So you've got the hydroxide ions and the hydrogen or hydronium ions. Those are charged, so they can get in between the other charges and interrupt those. Another thing would be detergents. Often you use these in lab on purpose because you want the proteins to denature. And they have a similar function where they just get in there and interrupt those weak bonds. Okay, so now on your single sheet of paper, you have a graphic organizer with the basics of proteins.